I'm Aiko Thompson, Associate Editor of the journal Topics in Spinal Cord Injury and Rehabilitation. And I'm a professor at the Medical University of South Carolina. I am super excited to introduce the presenters of today's webinar, Dr. Scheuren and Dr. Jones, who will talk about one of the journal's most read papers entitled Exploring the Landscape of Biomarkers in Spinal Cord Injury. Dr. Paulina Scheuren is a postdoctoral fellow at the International Collaboration on Repair Discoveries, known as Aiko, and the University of British Columbia in Vancouver, Canada. She earned her master's and PhD degrees in health sciences and technology at ETH Zurich and the SCI Center at Valgrind University Hospital. Her research is focused on developing mechanistically grounded biomarkers to elucidate disease mechanisms, predict recovery, and optimize the design of clinical trials in spinal cord injury. Her work is supported through multiple prestigious fellowships, including the International Foundation for Research in Paraplegia, Swiss National Science Foundation, Canadian Institute of Health Research, Michael Smith Health Research BC, as well as grants from the Wings for Life Foundation. Dr. Linda Jones is an associate professor at Thomas Jefferson University and a consultant to biotech companies, universities, and nonprofits, including the Christopher and Dana Reeve Foundation, to advance spinal cord injury research for over 20 years. After focusing on physical therapy interventions in the intensive care unit, she managed the first cell-based trial in SCI, followed by the first study utilizing cell-derived from human embryonic stem cells in the United States. She then joined the Craig Nielsen Foundation, managing the translational research portfolio and initiating and overseeing special projects. She is a co-chair of the Spinal Cord Injury Outcomes Partnership Endeavor, SCOPE, the former co-chair of the Spinal Cord Injury Outcomes, the co-chair of the American Spinal Injury Association Research Committee, and has served on NINDS SCI Common Data Elements Committee. Her interests lie in clinical trial design, outcome measures, and bridging gaps between preclinical and clinical research to promote better alignment between animal and human research. So before handing this over to Dr. Scheuren and Jones, I would like to remind the audience that we will be using both chat and Q&A and raising hand functions in this. So all the comments and questions will have a chance to be addressed after the presentation is over. So please hold off your questions until the presentation portion is done. Now, without further ado, Dr. Scheuren and Dr. Jones, please take it from here.
Oops, let me unmute. Can you see my screen? Hello?
Yes, we can.
Okay, and
with notes.
Yes.
Okay, correct mode?
Yes.
Okay, fantastic. So great to see everyone today. Although we can't see you, we know you're there. Paulina and I would like to thank you for the opportunity to present a summary of our biomarker paper, which was derived from a workshop that was sponsored by SCOPE. SCOPE is an Asia affiliate. So the order of presentation is I'm going to begin with a SCOPE overview and followed by general terminology on biomarkers. And then Paulina is really going to present the meat of the paper in terms of applicability and examples in spinal cord injury. And just to note, neither Paulina or I are experts in biomarkers. We are providing this paper, which is an overview. A number of our co-authors are experts. So if there are questions that we are unable to answer, we will be happy to get back to you after the presentation. And I cannot, there we go. Okay. So a bit of background on SCOPE. So as mentioned, okay, hold on just a minute. SCOPE was founded in 2006 as an academic industry agency partnership to advance the development of clinical trials and clinical practice protocols and validate therapeutic interventions for spinal cord injury. As mentioned, Jane Shea and I are the co-chairs, but I'd like to acknowledge our founding co-chairs, Dr. Andy Blight and John Steeves. So SCOPE is made up of partners and members. Our partners represent industry and nonprofits, as well as a CRO. Industry representatives are AbbVie, NerveGen, and Lineage Cell Therapeutics. Our nonprofits are Lineage and Wings for Life, and DP Clinical is a CRO member of SCOPE. We also have, or partner, I should say, we have approximately 30 members who also represent industry, academia, and nonprofits, and you can find the full list of our members on our website. So what are some of the things that SCOPE does? So one thing we do along with Asia is SCI Trials Finders. And if you haven't looked at this website, I would encourage you to do so. We take postings on clinicaltrial.gov on clinical trials, and we have an expert curation committee who takes these postings and makes them, derives them into understandable lay-friendly language for individuals living with spinal cord and their families. This website is also searchable, so you can search under the type of injury you have, your location, chronicity of injury, etc. Some of our recent publications span from data safety monitoring boards and relevance in spinal cord injury, patient-focused drug development, which is an activity to learn about what's important to people living with a given condition. And this was done in collaboration with NASCIC. This white paper can be found on our website, and a clinical trials update paper that was derived from another pre-course. Our recent workshops focused on MetaView on spinal cord injury research. What can we learn from how we do research? And a lot of this information was actually derived from SCI Trials Finder. And we recently had a workshop on the placebo effect and its impact on trial design. And you can look forward to a workshop at Asia 2026 on discomplete injury, how it's defined, and relevance in spinal cord injury clinical trials. So, as mentioned, the paper was derived from a workshop, and this workshop was a pre-course in 2024, and we had a very expert planning committee, extensive and expert planning committee. You'll recognize a number of these faces from SCOPE or Asia, but there are some new faces here as well. And I'd like to point out that we really had a range of expertise in our planning committee. So on the lower left, you see Dr. Chow. She is a pharmacologist ranging to Dr. Field-Fote with expertise in rehabilitation. So a wide range of expertise. These are the speakers and moderators who are also co-authors on the paper. And again, you'll see people that you recognize and people that you don't recognize. We had people with no expertise in spinal cord injury. And the order of the images here is by order of presenter. So as an example, Dr. Leptak from Eloquent was formerly with the FDA in the Agency for the Development of New Drugs for 14 years. So he doesn't know as much about spinal cord injury, but he brought his lens to spinal cord injury. And then we have people that we do know, such as Dr. Kwan and Dr. Park, who are themselves establishing biorepositories, which is really needed to advance the field in terms of biomarkers. Dr. Kleitman gave an excellent summary of the day, which really helped to inform the manuscript. So what is a biomarker? What is the definition? So the FDA definition is that it is a characteristic measured as an indicator of normal or pathogenic biological process or biological responses to an intervention. It is not a measure of how an individual feels, functions, or survives, which is often the focus of a phase 3 or pivotal trial. So that is the FDA definition. And now I'm going to talk a little bit about classes of biomarkers. Now, Dr. Scheuren is going to go into more detail about classes of biomarkers, but this is a lovely image that she created for our publication, which I think really nicely highlights different classes of biomarkers. So we have histologic and molecular biomarkers derived from tissue samples, tissue and blood samples. We also have radiographic, where in spinal cord injury, we often think about MRI. Physiologic, an example might be electrophysiology, which Dr. Scheuren is going to address later, and digital biomarkers, for example, sensors. So, sorry, my slide advancing is not working very well. There we go. So what do biomarkers measure? So orienting you a bit to this slide, the left side of the screen are biomarkers that are most relevant at the onset or progression of a disease or condition. The right side of the slide are biomarkers that are most relevant during the intervention phase, for example, in a clinical trial with a given intervention. So I'm going to go through these. And again, Paulina will expand upon these in her portion of the presentation. So if we start with, on the left side of the screen, susceptibility or risk biomarkers, this is the risk of developing the disease. Now, this is less relevant in spinal cord injury, but is highly relevant for diseases with a genetic component. And then we have prognostic biomarkers, which indicate the likelihood of disease progression. So in spinal cord injury, an example is our understanding of natural recovery. How does the disease or the condition progress, in our case, improve and or deteriorate? And diagnostic biomarkers, so indicative of the presence or absence of the disease. And then if we look at the line between the disease itself and intervention, we then have monitoring biomarkers that evaluate the status of a disease or condition. And then moving into the intervention phase, we have predictive biomarkers, which look at an individual's likelihood of a favorable response to an intervention. So what are their characteristics that would cause them to respond or not respond to a given intervention? And we also have response biomarkers. So what is an individual's actual response to an intervention? So predictive is what we think will happen, and response biomarkers is actually what did happen in a given individual. And then we have safety biomarkers, which are used, we use in clinical trials to look at adverse events to a given intervention. So why did we hold this workshop when we did? What was our rationale? The FDA has a biomarker qualification program, and to highlight, there are no qualified biomarkers in spinal cord injury. So this doesn't mean they won't accept a biomarker, but we haven't had a biomarker that was ready to go through the qualification program. Unlike brain injury, traumatic brain injury, which does have a couple of biomarkers that have been qualified for the FDA. And something that recently came out, actually within the last week from the FDA in February 2026, the prior standard for pivotal trials was typically one robustly executed clinical trial with a second confirmatory trial. Now, there were some situations where there was not required, for example, in rare diseases, but this was the general standard. But this has now changed, where the FDA is allowing one robust clinical trial with confirmatory evidence. So this could consist of a single trial supported by additional data such as mechanistic insights, real-world evidence, natural history studies, or animal models. And particularly when we think about mechanistic insights, biomarkers may actually play a role in this new approach to clinical trials. We don't know yet, it's too soon to know, but they may. So another part of our rationale is you'll all be very familiar with the INSCI exam, which all of us use routinely for many different reasons. We use it for diagnosis, for prognostication, for prediction and safety monitoring. But as we also all know, this is a very crude tool, particularly when we think about using INSCI as a prediction model. If we think about, let's say, using age impairment scale grades as a prediction, if we look at age impairment scale C, there's a broad range of outcomes in age impairment scale C, from minimal motor function potentially to walking. So this is really too crude to truly be used as a predictive tool, which is why we really need biomarkers. So another reason that we sponsored the trial when we did is there are emerging types of novel trial designs. I'm going to highlight one here, and I need to explain what it is before I can elucidate the impact of biomarkers or potential impact. So platform trials are a trial that is different. So our typical trial, we give an intervention and we observe what occurs through the clinical trial. We go to the end of the clinical trial and we determine the outcomes of the clinical trial based on the findings at the end of the study. Platform trials are trials that run multiple interventions in parallel. So there's a standardized control group and a standard of care that is given in conjunction with multiple interventions. And you can imagine that this might save time and efficiency in running clinical trials. The challenge in spinal cord injury is that our outcome measures are often evaluated, our endpoints, at 6 and 12 months, which really decreases the efficiency of a platform trial. Because what one can do in a platform trial is one can evaluate whether a given intervention needs to be dropped. So for example, if it's not efficacious, you would drop that arm and you might add a different intervention. But if we have to wait 6 or 12 months for the outcome measure from individuals within the trial, that would really extend the time frame for a platform trial. Whereas if we had an earlier biomarker that could be used in an interim analysis, this might really save us time and efficiency. So another reason that we sponsored the workshop when we did was really looking at the industry perspective, because a lot of our trials are sponsored by industry, also a lot by academia. But in looking at where biomarkers are used in the drug development pathway, kind of on the top portion of this image here, they can be used as markers of toxicity early in the development of a drug. They can be used as markers of target engagement. So given a drug, does the drug interact with the intended target? And if so, is that target modified and how much? And a biomarker can help us better understand this. Also, pharmacodynamic and pharmacokinetic biomarkers, et cetera. And I think another thing that's really important to appreciate from this image is that ideally our biomarkers are conserved across species. So we want a biomarker that is relevant both in our animal models and our human models, ideally. This may not always be possible, but this is something we have to consider in the biomarker development pathway. So from here, I'm going to turn it over to Dr. Scheuren, who's going to talk about relevant examples in spinal cord injury.
Okay, thanks, Linda. Just getting my screen set up here. Okay, I hope you can see everything. If not, please let me know. Yeah, so I'd also, at this point, before I begin, just like to thank the organizers for putting these webinar series together, and particularly for inviting us today to talk a bit about our recent paper. And as Linda already, she really touched on the main rationale of the pre-course and also the manuscript. And now I'd like to talk a little bit more or explore this in a little bit more detail, particularly in the context of spinal cord injury, give some examples, and also how these might be useful in clinical trials. So to start, I'd like to focus a bit on some of the molecular biofluid-based biomarkers that have been investigated in the field so far. So here we often refer to samples that are acquired either from the cerebrospinal fluid, so short CSF, or from the blood, so either serum or plasma, for example. And I would say the most promising biomarkers, at least in the context of acute spinal cord injury that have been investigated so far, include neurofilament light chain, so short NFL, which is a marker of neuronal damage, and also glial fibrillary acidic protein, GFAP, which is a marker of structural damage to astrocytes. And these are also quite common biomarkers in the context of neural trauma in general. So for example, GFAP is also part of an FDA-cleared test panel for traumatic brain injury, which makes it a quite interesting candidate for spinal cord injury as well. And obviously, there are also some other structural proteins or inflammatory cytokines that have been investigated and shown to be elevated after acute spinal cord injury. And unfortunately, I won't have time to go into all of these today because we're going to talk about many different types of biomarkers, but I just wanted to give an example of some more recent findings of how these types of markers can be useful for the diagnosis and prognosis in spinal cord injury. So this is a study that was published in 2023, and this is from work from Brian Kwan's lab here at Aiko, led by Sophie Stukas, where here you can see levels of NFL, so neurofilament light concentrations, and they measured these quite early after injury. So here starting at 24 hours after injury. And you can see quite clearly from this image that they really classify injury severity. So we see these elevated levels in AIS-A compared to AIS-C patients, for example, highlighting the diagnostic value of, for example, NFL as a biomarker in spinal cord injury. And importantly, they also here looked at the prognostic value. So here on the top panel, again, you can see NFL concentrations at these early time points. And then here showing that these are elevated acutely in those that do not convert in terms of their AIS score. And also here in the bottom, looking at the difference between those who end up motor complete or end up motor incomplete at follow-up. I think this was 6 months. Again, seeing these elevated responses in the more severely affected patients. So again, highlighting the use of some of these markers, not only for diagnosis, but also prognosis of spinal cord injury, and that they are related to the outcome at follow-up. And I think this nicely highlights the objective value of these markers in addition or in conjunction with clinical assessments. And while I think some things that here I'm just showing an example of, for example, NFL in the blood, they also looked at this for GFAP where we see similar findings. But these are also have also been investigated in the CSF. And important to mention is the concentrations in the blood and the CSF are actually correlated quite nicely. So while the magnitude is much higher in the CSF, these are actually quite well related, making blood also a very interesting biofluid to analyze, which also might be more clinically accessible and translatable. So here I just wanted to highlight the use of some of these biofluid markers for the diagnosis and prognosis of spinal cord injury, but what about their use in, for example, clinical trials? So here I just wanted to highlight some recent examples from recently completed clinical trials. And here's an example from the riluzole in spinal cord injury, a RISCUS study, and particularly the pharmacokinetic sub-study, which was led by Diana Chow, who was also a speaker in our session. And here they showed quite nicely that during the treatment administration, they found that the riluzole treated group showed this blunted response in phosphorylated neurofilament heavy chain, which similarly to neurofilament light chain is a marker of axonal damage, which was reduced compared to the placebo group. So really highlighting this biological response to the intervention. So highlighting the use of these markers as a pharmacodynamic or response biomarker in spinal cord injury clinical trials. Another interesting example is from also a more recently completed trial, the AXR 204 trial. This was in chronic cervical spinal cord injury, where they administered soluble NOGO receptor decoy at these chronic time points. And the interesting part of their biomarker analysis here was that the CSF proteome analysis showed a strong upregulation of, for example, here RTN4R, which is a component of the AXR 204, again demonstrating this target engagement in the broad sense that the decoy is where it's supposed to be. So Linda mentioned this a little bit when she talked about what these different markers measure. So this is an example of using these markers to prove target engagement. And what they also found is a prominent decrease in synaptic adhesion proteins, which potentially reflects stabilization of new synaptic structures, again, supporting the proposed mechanism of action, so this response biomarker. So now I'd like to, I talked a bit about some of these more molecular biomarkers derived from these different fluids, but I'd like to jump into another category of biomarkers, so-called histologic markers. And I would say that there are several reasons to explore the systemic immune system as a source of potential biomarkers in the field of spinal cord injury, both acute and chronic. The first and most obvious would be their potential use as susceptibility or risk biomarkers, so risk of infection or risk of developing, for example, secondary complications. As we know, this is one of the leading causes of death and rehospitalization in spinal cord injury. And there's also studies showing that they correlate inversely with recovery after injury. And the second would be monitoring biomarkers. So monitoring, for example, acute or persisting inflammation occurring within the central nervous system. And this is mainly derived, I would say, from CSF samples as well. But there's also some interesting postmortem studies, and this is one example here from 2006, where they looked at postmortem samples of spinal cords and saw that different cell types, for example, here microglial T cells, actually persist over time. Some are presented more acutely, others may persist. Again, highlighting the use of some of these histologic markers to monitor inflammatory responses over time after injury. And last but not least, there's also some studies showing their value, their prognostic value in the context of spinal cord injury. So, for example, the use of early white blood cell fluctuations to inform prognosis, or that they are related to recovery after acute spinal cord injury. So now I'd like to move on to a sort of a different category of biomarkers. So moving away from molecular and histologic markers to electrophysiological markers, or circuits and systems, as we named it in the paper. And I'd like to start with showing some data, some very recent data on the use of electrophysiological outcomes and how they might be useful in the context of clinical trials in spinal cord injury. So what we know is that electrophysiological outcomes help us detect, track, and quantify readouts of nerve function, including signal conduction or neural processing. So we know that they have this diagnostic and prognostic value in spinal cord injury. And one particular technique of interest would be, for example, here, so-called somatosensory evoked potentials, short SSEPs. And this is a electroencephalographic response, so a brain potential that is recorded in response to electrical stimulation of a peripheral nerve. So, for example, the tibial nerve or the median nerve, and it's a very classical neurophysiological technique. And what it allows us to do is to assess the functional integrity of these long tract spinal pathways within the spinal cord, particularly the dorsal column pathways, and how they might be affected or spared after an injury to the cord. And this marker was actually also included in a recent clinical trial, the so-called no-go inhibition in spinal cord injury clinical trial, which I was also personally involved in. And this marker was included in order to assess the integrity of these pathways, and then to understand to understand the relationship to administered treatment in these different subgroups of individuals. And what we found was that these electrophysiological outcomes were actually quite predictive to identify who will respond best to intervention. And I'll walk you through some of the data here to highlight what we found. So first of all, here you can see in individuals that have abolished responses, so no integrity of these pathways, assessed with this electrophysiological marker. And here is just showing a recovery over time from baseline to 6 months after the intervention. And then we're looking at recovery of skin self-care. This was all in cervical spinal cord injury. And then we see that there's no difference between the placebo in blue and the treatment group in yellow. However, if we move on to this group here of participants who have these preserved responses, we see that there's quite a bit of difference between the treated group in yellow and the placebo group. So improved recovery in those that have these preserved responses and are administered the drug. And most importantly, this difference actually also translated into a large effect size. So it's a very big difference, and there's hardly any overlap between our two treatment groups. And this is really important when we compare this to, for example, classical clinical stratification methods. So, for example, looking at subgroups of participants with motor complete versus those with motor incomplete spinal cord injury, where we did find in the same trial that those with motor incomplete injuries who received the drug showed improved recovery. However, this only translated into a small effect size. So these findings really highlight the use of these electrophysiological stratification methods to improve clinical trial design, and that they can really help us identify who will respond best to a given plasticity enhancing intervention. And most importantly, this also has substantial implications for the design or pragmatic implications for the design of future clinical trials. And I just wanted to give an example here. So what we did here is this is a classical power analysis. So this curve basically represents the 80% power curve. And here on the x-axis, you can see the effect size, so ranging from small, medium to large. And then here on the y-axis, you can see the total required sample size to achieve 80% power. And you can see here for the clinical stratification method, we would still need 120 participants to achieve 80% power, and we still only have these small effects. However, if we would only include participants with these preserved electrophysiological responses, this number would be reduced dramatically, so we'd only need 32 participants to achieve 80% power, and we also see these large effects. So this is really important in the context of planning and conducting clinical trials in spinal cord injury, because as we all know, recruitment is an ongoing challenge, and studying sample size is always challenging. So importantly, we did not only show that it could reduce the sample size, but it actually also reduces the enrollment period. So for this stratification method using these electrophysiological markers, we see that the trial would only take 33 months, whereas if we use this clinical stratification method, it would take up to 74 months. So this is a dramatic decrease in the time needed to recruit your participants for your trial. So I hope that with these results, I was able to highlight the use of some of these electrophysiological biomarkers to enhance clinical trial stratification. And I believe that this might have valuable implications for future clinical trials as well. So now I'd like to move on to some other electrophysiological outcomes that have been quite interesting in the field as well. And one such example is motor evoked potentials and their relationship to spasticity. And this is mainly work from Dr. Monica Perez's group, where they were primarily interested in motor complete spinal cord injury. And what they found is that in, for example, 60% of these participants, they found that they presented with spasticity. And then in these groups, they then performed electrophysiological outcomes, in particular MEPs, motor evoked potentials. So this is done using transcranial magnetic stimulation and then recording at the peripheral muscle. And what they found is in participants who showed spasticity, so that is all these red dots here on this side, these people also showed MEP responses, so residual corticospinal connectivity in those with spasticity, which was not the case in those without spasticity. So really highlighting these this residual connectivity in muscles that are below the injury after subacute and chronic motor complete spinal cord injury. And then some follow-up studies from the same group also showed the prognostic values of some of these markers and that they are related to motor recovery or AIS conversion. So here I'd like to focus your attention to this side at first. So those with spasticity and MEPs actually showed a subgroup of, or the majority of these participants improved in terms of the AIS grade, and they also improved in terms of motor skills, so moving here from these white dots to the triangles, which was not the case on this side here, so those without spasticity or without MEPs. So here really just highlighting the potential use of these markers as well for the diagnosis and prognosis of spinal cord injury. So now I'd like to move on to other types of markers that we can use to assess circuits and system, and in particular, imaging biomarkers. So there's many studies looking into this. I just highlighted a few here, and we know that imaging has a strong diagnostic and also prognostic value. So for example, looking at the cross-sectional area of the cord or looking at spared tissue within the cord, which is termed tissue bridges, which has also been related to recovery after spinal cord injury. And interestingly, recent findings from a study led by Lynn Fana in Patrick Freund's groups at Belgris in Zurich, we showed that some of these MRI markers may actually be useful to detect response to intervention in a secondary analysis of the no-go inhibition of the NISCI clinical trial. And here you can see on the y-axis the percent change in cross-sectional area over time. You can see in the placebo group this decreases, so we have this ongoing degeneration, which has been reported in the literature, which doesn't seem to be the case in the NG101 treated group. So again, highlighting that administration of NG101 might slow post-injury degeneration in this treated group. So really highlighting the use of some of these imaging biomarkers, not only for the diagnosis and prognosis, but also potential response to intervention in clinical trials. So now I talked a lot about biomarkers in the context of motor recovery, but I'd like to focus a bit of our attention on some of the other systems that we all know are very important after spinal cord injury as well. So for example, here, the autonomic nervous system, so both the sympathetic and parasympathetic nervous system, which we know can be quite disrupted after spinal cord injury as well. So why would we even be interested in studying biomarkers in this system? I think the reasons are quite obvious. So they are really listed among the highest priorities by the community. They have a devastating impact on the quality of life, and are the most common cause among the most common causes of morbidity after spinal cord injury. So it makes sense that there might be a potential use of some of these cardiorespiratory biomarkers to, for example, identify individuals who are at risk of developing some of these cardiac or respiratory dysfunction after injury, and who would then benefit most from an early intervention. And I just listed some examples here that also came up during the pre-course. So for example, looking at heart size, blood pressure, or forced vital capacity, which have all been investigated in the context of spinal cord injury. And I think important to mention here is also that there are many ongoing clinical trials looking at improvements in autonomic function, where so biomarkers in this context would be of great interest as well. So to conclude a little bit, I hope we were able to convince you that biomarkers can be quite useful in spinal cord injury, and also particularly in the context of spinal cord injury clinical trials. So an example being the use of electrophysiological markers to improve participant stratification, so to enhance predictive enrichment strategies for future clinical trials. They can help us inform pre-planned subgroup analysis. So for example, define specific subgroups in advance that you will include in your trial. For example, those with or without spasticity and how they respond to a certain intervention. To guide interim analyses. Linda already mentioned this in the introduction, in the context of platform trials or different types of adaptive designs where biomarkers are actually an integral component of some of these designs. Also, for example, the use of CSF or blood markers to confirm target engagement. Or lastly, another example, the use of MRI markers or CSF to detect response to intervention. So I'd like to just end here with some considerations for spinal cord injury on kind of this pathway to biomarker development. And as Linda said, there is currently no biomarker that has been cleared by the FDA yet in the context of spinal cord injury. But some things to consider on this path would be, first of all, early engagement of regulatory bodies. So it's important to establish clear timeline, metrics, and goals. The the use of analytical and clinical validation to ensure specificity, accuracy, and precision, and to make sure that these biomarkers are actually doing what they're supposed to do. So this kind of gets back to the context of use. And here, it's really important to consider retrospective analysis and also prospective analyses in order to validate some of these biomarkers. Adherence to best practice is quite an important factor. So here, what I mean is that researchers and healthcare professionals should utilize available infrastructure and resources to ensure that they, for example, meet the standards for data management, protocols, etc. And last but not least, coordination of multiple stakeholders. So given that there is a substantial time and financial resources that are needed to advance biomarker from development to qualification, it's really important to form consortia that include both industry partners, academic institutions, and, for example, disease foundations, and that this might represent the most effective long-term strategy along this path. So with that, I'd like to thank you all for your attention. Again, I'd like to thank the organizers and the planning committee, as Linda nicely introduced, and all the speakers and moderators of the session. Thank you.
Thank you so much. That was absolutely wonderful. It's great. So while we wait for some questions may come in, if you guys are okay, I would like to go ahead and then start with my questions. And so one of the things listening to your presentation and reading your fantastic paper, I thought about is like more like a personal question. So I want to know, what was the most exciting biomarker discovery or development that you saw over the past couple of years? And if you could give me like the example, like each of you could give me some example, that would be great. But that's really inspiring for us to hear what you saw. So if you could, maybe you can start, Paulina.
Sure. So I think I would say in general, one of the most exciting things, without like singling out a specific biomarker at start, would be just the fact that people are using biomarkers more and more, both in clinical studies and clinical trials. We see all of these recent trials included some type of biomarker or even multiple types of biomarker, where there's multiple sub-studies investigating all these different types of biomarkers and their utility in the context of spinal cord injury. Yeah, I would say that that's probably the most exciting part around the biomarker topic. And personally, I'm obviously very excited about our own findings in terms of the electrophysiological outcomes, but that's a bit of my bias flowing into that for sure. Yeah, that we finally have these tools where we can really improve the design of clinical trials using these different markers or potential tools that are also now going to be integrated into future trials. Linda, if you have anything to add, feel free, but
No, I would agree. I think the most exciting thing is that we are seeing biomarkers used in different ways in clinical trials, particularly in terms of pharmacokinetics, which drives trial design, identifies potentially characteristics of responders, et cetera. And those data are being used for, let's say, the next stage of a clinical trial. If the drug doesn't get to the target, then it can't work. So looking at those kind of factors is really important, in addition to electrophysiology, neurofilament, and imaging advances.
Yeah, absolutely. I was actually impressed. I didn't, because I'm also using electrophysiology myself, and we don't think about SCP as the biomarker, and then your study really shows the utility of it. It was quite remarkable. And as Linda mentioned, the target engagement can be tested with biomarkers. It's absolutely fantastic. It was very, very educational to me, and I'm pretty sure for the rest of the audience felt the same. It's a great summary and a great presentation you guys gave. So the other thing then I wanna here, maybe I was expecting a little bit more about. Maybe you didn't have time to touch on with this limited time frame, but which you also but mentioned in a paper is the somewhat more of a collaborative effort in this biomarker, the development, validation, discovery, all of that. Could you give us some examples of collaborative effort that can facilitate or accelerate the process?
Should I go ahead, Linda?
Sure.
So, yeah, thanks for that question, because as you highlighted, we were not able to go into all the details or mention everything in the presentation. So I'm happy that we get to, and it's a very important question. So I would say that probably one of the, like what I can think of now as the most important collaborative efforts, for example, in the context of some of these biofluid-based biomarkers and also other type of biomarkers would be, which is already happening in the field. So the establishment of biobanking systems, which are in different places around the world being established in the field to really facilitate biomarker research, biomarker development, and hopefully biomarker qualifications eventually. And along similar lines, I would say also the broad use of some of these samples and data that is being stored in some of these biobanks. Because, yeah, for which will be important for example, I talked a little bit about this in my last slide, the analytical and a validation of some of these approaches, I think would be very important. And I think, yeah, in general, I would say that it's quite a bit of an effort if we, as a field, to move from biomarker development to qualification eventually. So I don't believe that this is something that, for example, a single academic lab can achieve, or a single biotech company, for example. And that's where these pre-competitive biomarker consortia become very important to move this forward in a more effective manner, I would say.
Yeah, I would add to that and just say, I think the real collaboration is between the investigators and also potentially the funders in terms of targeted funding for biomarker development. Biomarkers have been something we've been talking about at least since I entered the field and are still talking about. We're making great progress, but working together, collaborative efforts really facilitates things. And as Paulina mentioned, even just the mechanics of how samples are collected and stored and trying to implement certain standards by which that occurs will help us. It seems pretty basic, but will help us develop as a field. I also understand that Wings for Life has an initiative where they are identifying the biorepositories throughout the world and what they contain. That'll be really helpful, so you can at least see where samples are and what they consist of. So those are areas I see where, as a field, we can collaborate to advance our understanding of biomarkers.
Yeah, that's absolutely great. We just don't think about it too much, which kind of leads to the another thing I want to ask about. So like both of you mentioned earlier in this, like doing this by a single lab or a group is really hard to go any further. So, but if each one of us, if we want to contribute to this biomarker discovery, development, or implementation, like what can we do as investigators or lab, like in our practice? What would be your recommendation?
Very good question.
Like, should we try to go up? Like as Linda mentioned, there will be like some repositories, and then should we try to like contact them and try to contribute by sending some samples, or try to maybe so how can we contribute? Because clearly this is really important, and many of us, well, at least me, we don't know how to do it. So if you could give us some sort of like a guidance there, like how can we contribute?
Yeah, I mean, the example you gave is perfect. For example, contributing samples, if you have samples, sharing data, I think is a huge, and I think this is really something that the field is moving towards more and more in the last several years. So this is already happening, I would say, at a certain level, which is great, and just continuing to do that. I think, yeah, one important part is also considering like in the generation of that data is to really ensure reproducibility and also robust biomarker data when it's being acquired, for example. So sticking to some of these best practices would be, for example, another example. Linda mentioned, and maybe she can expand on this a little bit as well, the funders aspect in the previous question. I think that kind of comes in here as well. So really kind of being aware of some of these initiatives that are like as for example, an academic scientist, a research lab, being aware of some of these initiatives and contributing to them that are, yeah, at different levels.
Yeah, I think data sharing is an aspect of our field that's really blossoming right now. We have a dedicated repository for data sharing in the Open Data Commons for spinal cord injury, which is the only dedicated spinal cord injury repository for data sharing. So sharing our data related to biomarkers is important. Paulina mentioned contributing samples to various data sets. I don't know what the thresholds are exactly, but as an example, Brian Kwan is running a large biobank repository. Andrew Park also has something that's more local at Craig Hospital, but contacting those investigators to see how you might contribute, I think would be really helpful. And as Paulina mentioned, from the funding perspective, I think that, to my knowledge, at least, most of the funders are open to submissions on biomarkers, but perhaps calls that really focus on that development could be helpful.
Yeah, absolutely. It's great. I think there are some questions coming in, and then luckily you just answered some of the questions. Some of the audience question was about the availability of those repositories in the SCI field, and you just answered so there are a few other questions, but I also realized, I don't know if we have time to go, because it's just almost about the time.
We
Maybe
have time.
okay. Then there is the one question that I think it's just came in a few minutes ago about SSCP. I cannot pronounce the name right, but Mr. Dr. Rapp, if it has a question, if still online, could you join and then ask the question about the biomarker SSCP? Could you add the person?
I think in case it's not working, I can see the question here. So this was
Okay.
from Rüdiger Rupp asking about SSCP and whether or not it actually that it reduces the number of participants, but not necessarily the enrollment period of the study, and how it can be useful in this context. So neuropotential studies. Oh, there he is.
Yes.
I'm
Great.
not sure if he can turn on his mic or not.
I try to do my best. I'm very sorry
Perfect.
for that. Very sorry for that. So first of all, great talks and really wonderful insights. So my question was related, and then Paulina, we had these discussions before. So for me, actually, a biomarker is an additional inclusion criteria, and it might actually then that you end up with less participants actually then that you need for enrollment in order to show a statistical effect of a drug or of any kind of intervention. But the question for me is, and we all have these problems actually then in recruitment, does it really help then to reduce also the time then of a clinical trial? So are we then able then to speed up processes? I mean, that would be particularly when we have now these concurrent studies actually then and well, a lower number of patients, at least in industrial countries. That's an important, I would say, and it would be an important advantage than of including a biomarker in the trial. And this is something maybe you have investigated in more detail. Sorry for my long.
No, great question. I think it's a very important question, and I'm happy to elaborate on this a little bit more. Because based on our analyses, and I mean, obviously, I can only talk for the NISCI trial and that specific sample, but there I can say yes, that it also would reduce not only the number of participants, but also the time needed to recruit these participants.
But that's
which,
a great
as you
news.
said, is probably more important than the actual number, right? Like, because
Yeah,
Linda
exactly.
mentioned this when she introduced platform trials. Like, we need faster trials. We need not only faster trials in order to succeed faster, but also to fail faster, right? To know, is something actually not working? So, like, if we can move up our interim analysis and use a biomarker to test a certain intervention early in a specific subgroup, for example, based on our biomarker stratification, that would greatly enhance the efficiency of all clinical trials and not just so based on the analysis that we did for the NISCI trial, I can say yes, the data says it reduced. It actually would have been shorter than the NISCI trial itself. But obviously, this will always change a little bit. And I mean, the NISCI trial, as you know very well, because you were a part of it, of course, it is embedded within the EMSCI network, which obviously has it's a huge network that has been established over many, many years. So this is like where we base our analysis on. And obviously this might change if you move into a single center study, for example. I don't have those numbers, but based on the actual recruitment rates that we saw within the NISCI trial, yes, it would also reduce the time.
Great. Thanks, Paulina.
That is absolutely wonderful. Because when I think of each participant's time and effort in coming into clinical trial, especially the intervention trials, and you point out exactly test fast, and then if it's to fail fast is really important, because those are the people who are living and then experience whatever they're suffering they're having. So this is really wonderful. So we have 1 more minute left, and then before we close this, thank you so much for this wonderful presentation, and then all the Q&A, and then covering the important points. Before we close this, we want to just remind the audience with the future announcement. If we could go back to the announcement of the future webinar scheduled in April. I don't know if you can. Yes, great. So the Azure TSCIR webinar series will continue. The next one is scheduled in April 8th, and the registration is open. It's going to be on a completely different topic, and we are expecting some very exciting presentation and discussion. So please remember to register for the next one as well, and wait to hear from us more. Thank you all so much. This has been really wonderful. Thank you, Linda, and then thank you, Paulina.
Thank you. Thanks for having us.
Thank you.